Hello, students, and welcome to this brief overview of how the neuromuscular junction and how neuronal transport in general works. You need to know this largely so that you can use it when we're talking about the different types of tracts and the transmission of information within the autonomic nervous system and the within the sympathetic and the parasympathetic nervous system. So hello, students. This is going to be a brief overview of how neuronal transmission works, so a little bit of an idea of what neurons actually are, and then how the information can go from one to the other, so that you can use it when we're talking about the autonomic nervous system, the sympathetic and the parasympathetic nervous system. So it's one of the things that we're going to be talking about relatively soon on in the semester after we go over a little bit of neural anatomy, but this is how this whole thing is going to work. Remember that in any type of situation, we're going to have some sort of neuron that's going to start out the whole process. And then that neuron, or whatever that may be, I mean, it doesn't need to be a neuron. It could be a photoreceptor cell. It could be a gustatory cell. It could be any type of electronic transducing cell. So something that takes information from one place and is pushing it on to the next place. That is going to... Whoa, okay, that's fine. It's going to synapse. So this is a synapse. And the synapse is a gap between these two different cells, but it's going to synapse with another cell. And let's say that this other cell is a multipolar neuron, and it synapses itself with a bunch of other different types of things. So remember, there's the different types of neurons. There's the bipolar neurons, which we saw a lot in the eye. There are unipolar neurons. There are also pseudo-unipolar neurons. And then there are multipolar neurons. And they all have an axon. So one of these is going to actually be longer than the other. So my little TIE fighter over here is not necessarily appropriately drawn. We can fix that though by going like that. Look, ooh, so there's a dendrite and an axon. Dendrites and an axon. Pseudo-unipolar neurons are a little bit different because they are a little bit different. So, in any case, we have this synapse. We have information that is going to travel from one place down, and it's called an action potential. Action potentials are a positive influx of ions. So we have something that makes it go positive over time. And that information is going to travel, opening voltage-gated sodium channels and voltage-gated potassium channels in a wave-like fashion all the way down the axon until it gets towards the synaptic knob or the synaptic bouton or whatever you want to call it, towards the synapse, where there will be a cell that will, or not a cell, a portion of the cell that has a voltage-gated calcium channel. And that voltage-gated calcium channel will see the voltage increase and open, and that will allow calcium to go into the cell, and that calcium is going to make its way to synaptic tethering proteins or snare proteins, and those will shorten until it releases the neurotransmitter into the cleft. So that calcium will bind to those tethering proteins. It'll make those tethering proteins shorten. They're going to bring their thing down, and when two different things that are made up of membranes touch each other, it'll release everything into the synaptic cleft. That neurotransmitter is going to be released into the cleft, and it can either be retaken up, broken down into its different components, or it will go and bind, or it could diffuse away, but it could also bind to a receptor on the other side of the cell. Now, depending on what the neurotransmitter is, it's going to bind to this receptor. The receptor is either going to be one of two types. It'll be a channel receptor. Channel receptors just open up and let in whatever they let in. If it's a sodium channel, then it will let in sodium. So that will be actually more similar to like the nicotinic acetylcholine receptor in the neuromuscular junction. So, but we have our neurotransmitter binding to the postganglionic cell, and then it will, in this case, it'll open. So neurotransmitters. So when that neurotransmitter binds, there's one of two things that'll happen. We largely talked about having a ligand-gated channel, so a channel receptor. These channel receptors will open the channel, and then whatever will go in or out. So if it's a potassium channel, that means potassium will go out, and it'll be inhibitory to this next neuron. So it'll either raise the voltage or lower the voltage. Or it could be a G protein coupled receptor, a GPCR, G protein coupled receptor. These GPCRs are going to be like some of the taste receptors. There'll be something that'll bind to it, but instead of that thing going through or something else going through, it'll send other messengers to do something. It might open up other channels somewhere else in the cell, or it might change the gene transcription inside of that cell to have something else happen. So those are the two different options that we have, and that's what this neurotransmitter is going to do. In this case, just because we can, we're going to say that it's a sodium channel, and that sodium channel is going to open, and then that means that sodium is going to go into the cleft. And if we have sodium outside of a cell, that sodium is going to go into the cell, and that's going to make it more positive. If we get enough positivity here at what is called the axon hillock, that is going to be the place where the first set of voltage-gated sodium channels are going to be. And if those voltage-gated sodium channels are there, they can open, and then sodium can go in, and then that'll set off the chain reaction until we end up getting all the way down to the end of our whole entire axon, and then the same thing happens again. That's neuronal transport. Now, if we don't have something that's positive here, remember, it has to reach positivity. It has to reach that threshold or the voltage at which voltage-gated sodium channels open. And if it doesn't, nothing happens. No action potential. Action potentials are all or nothing. So this is some of that, the way that we can get information to go from one place to another within the nervous system. Hopefully this helps. Hopefully it jogs a little bit of your memory. If it doesn't, if you're still confused about this whole thing, stop by and just ask. Oh, that was corny. Or whatever. Anyway, just let me know and I can try to help. We'll talk about this in class too.